Yeah, so the agenda for today's meeting is probably about an hour or so. I will go over some, uh, I guess, from basics to some recent literature in grid cells. So it's kind of a broad area. Um, and the rest of the time, Jeff has something to present about how movements are created, right? I don't think I'm remembering
Yeah,
the whole thing.
Which I
represented.
guess
Yeah.
happens to also cover grid cells. So that seems like
Right,
a
Yes.
fortunate
exactly. They're
coincidence.
very related, very related. And just to interrupt you before you even get started, OJ, this grid cells are one of the hardest topics I've ever dealt with in neuroscience. And
So I'll try
and
to do my best.
so if you find this confusing, well, then you're in, don't worry about it. If you don't find it confusing, then I'll worry about myself. But when I first really dove into grid cells, it was probably 2 months before I had a decent understanding of what's going on, and it was like it is really confusing. So if
Okay,
just to let you
great. Because
know,
in the middle, I wrote one sentence. I was like, "This is so obvious," and then I thought about it, and I was like, "This is very not obvious." And
well,
I'll
it might
point
be
it out when
easier
I
for
get
you,
there.
but it wasn't just me at Nementa. It was Subutai, myself, and a bunch of other people all just scratching their heads over this for so long. It's
Yeah,
unbelievable, but it's understandable. Okay,
yeah.
sorry. I just didn't want
All
to sort
right.
of preface that. It's difficult.
Yeah, no worries. And also yesterday I was mostly at urgent care, so like my brain is a little
Oh
bit,
no. You
yeah.
okay?
But yeah, I'm much better. Like just like a 24-hour food poisoning. Um,
Oh,
but like yeah, after sleep I'm so much better. So we're good to go, basically.
Yeah, wow. Yeah,
Okay, so
glad to hear you're feeling better.
yeah, I think that yeah, like yesterday night I was like seriously considering like, oh my goodness, like can I do this? Can I not do this? And then it was like a good night's sleep. Like I still haven't
mm.
eaten yet, but it's you'll see like Arthur is here because I have a banana, and he really wants that banana. But it's mine, Arthur. It's like mom's first food in 24 hours, so he cannot have the banana. Okay, anyways, that's too much rambling. We can just probably cut that part and start the recording from here, Will. Um, okay, so literature review on grid cells. The short table of contents. I will go over like a grid cell 101. I think things that I think most people can agree on, I would say. And then the middle portion I'll be dedicating it to re-anchoring of grid cells. So this is kind of it's still a literature review, but it's inspired from the fact that Nils and I are working on this implementation request for comments RFC on um, how to handle kind of re-anchoring in Monty and what to do with uh, off object observation. So that was part of the actual motivation for doing this grid cell literature review. And some recent work. So by recent, I mean like from 2022 and since then. So the reason why I also mentioned that year, okay, grid cell 101, is that there's actually, if you search for grid cells on YouTube, Numenta has a whole basically course about it. There's so many videos from the channel Numenta Theory that I wanted to like include. So a lot of them are like these kind of research meetings that are like an hour or an hour and a half. I did not watch all like 20 videos. It's just FYI. Um, and there's also I think one of the best resources for like getting up to speed is watching this 2014 Nobel Prize in Physiology of Medicine. So, the lecture has been, I think they record all the lectures and put it on YouTube, but basically this is like a 3 hour-ish lecture from the Nobel Prize winners, John O'Keefe, Maybert Moser, and Edward Moser on place cells and grid cells. But in general, so I think these resources are going to go, some of them are going to go into like much more detail, definitely, than what I will cover today. But I just thought that for people like me, I need something that is like, I wanted to make something that was like very accessible to the community for somebody who may not even be in systems neuroscience, even though this is, as Jeff said, like a very difficult topic to grok.
And it
So yeah,
goes also saying that we don't have to implement, I don't expect we'll ever implement the details here of grid cells in Monty.
that's right.
It's very, very detailed, cellular biology, and we don't generally want to do that if we don't have to. So
Yep.
if you want to
So it's
understand it, you have to
mostly for
crack
kind
it.
of inspiration about like what happens when grid cells do re-anchor. So we just wanted, well, I guess I was kind of curious on like what's the biological justification for doing something or doing this in Monty. So we don't need to follow the biological mechanism, but at least in the beginning, I wanted to know like uh, if there's any connection at all. Okay, so before I can even talk about grids, I'm going to just have like one slide on place cells.
I'm sorry, I'll keep
In the
interrupting, Hojae. I'm sorry about this, but it's more than that. I mean, many of the ideas in Monty we wouldn't have thought of if we didn't read about grid cells. The
yeah,
ideas of unique locations in space, the re-anchoring, how shapes, there's a whole bunch of concepts that all we knew is going into it was like, hey, we have to have some representation of location and therefore some reference. But we didn't understand path integration. We didn't understand this re-anchor. We didn't understand unique representations of location. So it's more than just like, oh, these are things we got from studying grid cells. So it was pretty important.
and those were
Yeah, it's more kind of I guess this is why we're revisiting this topic. Because as you say, Jeff, it underpins a huge amount of the Thousand Brains theory. But in terms of kind of why are we talking about it today, it's more because, we're starting to add, we're planning on adding certain properties to how the locations are updated in Monty, like things like re-anchoring, and kind of when you move off of an object, which would be analogous to like removing a mouse from an environment and things like that. So it just seemed worthwhile to just kind of revisit the literature, see what actual grid cells do in those kinds of settings. Does that like kind of correspond to what our intuitions were for what we would want to implement? Are they different? If they're different, is there something kind of interesting there to think about? So that's kind of why, I guess, Hojae's doing this particularly today.
Yeah, great.
Yeah, so like I said, I will just cover one slide about place cells. And I'm going to show you this video, but this video has no sound to it. But basically, what you just to orient people, what you're going to see is a mouse with an electrode implanted on its head that is running around this box square environment. And basically, you're going to see these kinds of like black lines as indicating the mass trajectory, and then some colorful dots where the cell, whenever the cell is firing. So.
And the different colors, I guess, correspond to different cells.
It looks like it. The original recording's only recorded from one cell, like you see in the red on the black.
Yes, yeah. I was going to say I couldn't find the original video. I think it was a little bit too far to go. But
all over. Yeah, doesn't matter.
yeah, so um, but the idea is very similar to what happened in the original video, except this one has like 10 simultaneous recordings going on instead of just one. And of course, like if you try to make like a common way that these are presented in papers is this firing rate heat map. So the size of the heat map is the same as the size of the room, and wherever you see kind of red is where there was a high firing rate of that cell. And blue is where there's less. And of course, it's for one cell, it's a little bit easy to visualize. For multiple, it's kind of difficult. I mean, it kind of blends together, but that's the general idea. So place cell seems to fire at locations regardless of any sensory input. So that was kind of so this was like telling the location, hence the name place cells of the animal. And I mean, this might seem like a random fact, but it's like, okay, cool, like we found this cell. But I think the fact that's like a little bit less appreciated is that it seems to be like one kind of sort of independent from sensory inputs. And this is what John Keefe kind of mentioned in his Nobel Prize lecture, is that it seems to be generated within the brain itself. And this was, I'm mentioning this because this was kind of the motivation for looking for grid cells in the first place. Oops. And let's see if we can go to the next slide somehow. Okay, there we go. So
But
if
Hojae, I
the,
think it's worth pointing out a few things here. If
Mhm.
you go back to the previous slide.
Yeah.
First of all, the cells always would fire in the same locations in the same environment. But if the rat perceived it as a different environment, the cells would fire in different locations.
Yes.
So it wasn't like these are the locations in a square. These are the locations in this room. And if you put a painting on the wall in the room and the rat felt it was a different room, a painted placard, then the grid cells would remap and it would be in a different location.
Yep.
I mean, the place cells would remap. And
Yeah.
the idea that they're generating the brain itself, so it does require visual input or some sort of input to know what environment it's in. But if you turn the lights off and the rat moves around in the dark, the place cells still fire, which is the concept of path integration. So at times you don't need sensory input. Once you've locked into this room, you know what room you're in, then the cells will continue to fire as you move about, even though there's no sensory input. But it's not completely independent sensory input because the rat has to determine where it is in the world.
Yeah. That's why it's like it's a little bit.
Okay, I just want to
Yeah.
add some flavor on top of that.
Yeah,
That's it.
definitely. I think we'll definitely also cover that and re-anchoring
sorry.
because no.
Yeah.
This is I
Right.
think I think I meant to say it.
That's all right. If you don't
My
want to,
brain
I'll
is
just
just
keep going.
like,
I'm just going to add color
yeah,
to your
please.
comment view. No, just keep going.
Yes. Yeah. Okay, I don't want this to keep playing. Okay.
So,
But it
quick
will.
question. Do these place
Yes.
cells have different scales, like we have in grid cells? Because I can see some cells are the firing range is like a larger scale, and others are kind of like smaller. Do we know if they have different scales or? Um,
Does
Yes.
anybody want to take this up?
Yeah, I
Yeah,
think
okay,
so.
Jeff.
I know that it's true in grid cells. I believe it's true in place cells as well. Whereas, as you go across, these are all in the hippocampal complex. And as you move across from one direction to the other, the scales of these things jump. I believe the place cells scale jumps with the same as the grid cells. I'm not 100% certain, but like 95% certain about that. But you don't see them you don't generally see them mixed up. Like the place cells and the grid cells in one little area seem to be the same scale, I believe. It's a bit misleading because you think, oh, this one cell just encodes this one space, but of course these cells are reused. And in larger environments, they can be active in multiple places, and of course they can be active in other environments. So it's like nothing in the brain is a single cell grandmother cell, and that's true here too. Although at certain scales and certain environments, it looks like one cell responds to this one location in this one environment.
Yeah.
Yeah, a good question. So this discovery of place cells in 1971, if we were to kind of follow the idea that, this is at least somewhat independent of sensory inputs and seems to be one generated from the brain itself, then where are these like place cells and how are they generated? So that was kind of the next natural question. And so, I mean, there's a lot of series of experiments that led up to this, but basically the idea was that instead of looking at, I think the original place cell was recorded in CA1 of the hippocampus. And so the Mosers, I think their graduate advisor, I think, was Perry Anderson. And then I think Perry Anderson, one of his work was trying to figure out the hippocampal, like entorhinal cortex circuitry. So, there is kind of a simplified version. I don't think this is actually the entire circuit by any means, but
Oh, sorry.
sorry,
I
go ahead.
I wasn't seeing your screen anymore, but I just switched to a different tab. Sorry.
Oh, okay, okay. That's fine. Um, so, basically there's some EC sensory entorhinal cortex, layer 2, layer 3, CA1, CA3, DG is for dentate gyrus. But if you look at a very simplified neuron, cell in entorhinal cortex from layer 3, there is a soma that projects this axon. There's a very simplified arrow, and the CA1 kind of captures it. So there's like it's called a monosynaptic pathway, because there's just only one synapse associated with it. There's some other pathways where like the layer 2 entorhinal cortex will project to the dentate gyrus, and the green dentate gyrus, will project to CA3, and CA3 will project to CA1. We don't need to go into this, but basically there's a couple of monosynaptic or trisynaptic circuitry where CA1 can be influenced from EC. And I think this is already I think this is established, but now we're finding out there's more connections to this. But the idea was like, okay, what they knew at that time at least was, okay, so if we want to know what where place cells are generated, let's look one synapse upstream at the entorhinal cortex, and do kind of the same experiment. We're going to stick some electrodes and see what we find. And
Just to kind of
and this is
comment, because I've not seen this kind of view before, but it's kind of interesting that the way the kind of apical dendrites are arranged in the hippocampal complex, it's almost like it's an inside-out cortical layer, as in because it's kind of like L1 is at the bottom, and then you have kind of almost the pyramidal projection from L6 is kind of superficial. But yeah, I don't know. It's just kind of interesting in terms of thinking about the kind of potential evolution.
I think at this point, I think it's just all things are flipped on top of each other. So
Yeah.
up and down doesn't make any sense anymore.
Right.
I think they'll. I'm not sure if it's the hippocampus or never mind. Or the entorhinal cortex, but I think their cell soma is like mostly in what they call layer 2 or 3.
Yeah,
If you look at
right.
like
So like the
microscopy images,
superficial.
yeah.
So no matter how you orient it,
Yeah.
it's kind of like it's closest to the edge of the structure.
And you want Jeff?
No,
were saying
I
same
just at this point, it's all topsy-turvy. So what's up and what's down doesn't make much difference. What this is all coming across the neocortical sheet. It comes around the side of the brain and wraps up into the middle, and it stops becoming classic neocortex, and it starts becoming wonky. So it's the same sheet, but so the first thing is the entorhinal cortex, which is okay. It's no longer 6 layers. They made up some number of layers, and then you can see it sort of wraps into CA1, then it wraps around underneath, and so what's top, what's bottom is kind of meaningless at this point, I think. And one of the theories that was proposed a long time ago, which I kind of like, but I'm not sure if it's true or not, and I've mentioned this many times, is that this circuitry where you have these three-layer tissues wrapping on top of one another could have evolved into the 6-layer cortex. So you can imagine this is how it started, and the basic properties that we assume in the cortical column are all here, plus some other stuff. And then evolution just guided, hey, well, instead of this wrapping us around on each other, let's make it a thing and extend it out, right? And then it becomes a 6-layer cortex. But I find these circuitry diagrams of the hippocampal complex are very, very confusing and hard to interpret, and different researchers say different things. And so I've generally come to say, yeah, don't worry about it too much. I mean, it's basic connectivity is important, but what's up and down, what layers are labeled 1, 2, and 3 are kind of meaningless at this point.
Yeah. So continuing on, doing the same experiment where the electrodes are stuck in, we can trace where the mouse has been with the gray lines and the black with that one particular grid cell where it fired. And the surprising finding was that, okay, this seems to be firing at like a this triangular or hexagonal lattice like pattern and grid cells. So I'm going to go over like a few properties, or I guess characteristics of, not really properties, but characteristics of grid cells that are like commonly used in commonly used terms, I guess, in papers. So like when we talk about grid cells, sometimes we say like, oh, the the phase is
I
off.
want to make sure I want to make sure everyone understands this, because can be, the basics can be very, very confusing. When we see this pattern like this is a single cell firing in multiple locations in an environment, whereas opposed to place cells was a cell that fired in one location in an environment. So I want that basic idea is that this is one cell we're looking at here. And of course, every location and environment, there'll be different grid cells or different place cells firing. So there's always some activity, and so here uniquely, grid cells seem to not be very specific. They do not represent a specific location. They represent multiple locations, and so they're not a very good indicator of location.
Yes.
Yeah, in this specific figure, the red and the green and the blue, they're different cells, right? Or so it's
Yes,
like
there's two cells.
Okay.
Yeah.
All right.
So there's
You're going to
two grid
explain
cells.
this? Are you going to explain this?
Oh yeah, you're welcome to take over too. So,
Sorry, it's hard for me to.
oh no. I mean,
It's hard
like,
for me to keep quiet.
no, I literally think that you can do a better job explaining. So that's why I'm not commenting on your frequent comments. I like it. So yeah, so in this case, these are two green grid cell and a blue grid cell, and in the phase, the same kind of periodic, like the same pattern exists, except one is just shifted from one another. So this is not an absolute measure. It's just it's a relative measure to like another grid cell. Scale obviously is when the spacing between the centroids or center of the firing fields are extended. So it's just spacing of the grid cells. And orientation, I mean, this is shown kind of relative. So like the blue grids. So if we say that the green one is kind of aligned vertically, then we can say that the blue is kind of rotated slightly, 10, 15 degrees. Usually it's orientation sometime, and this one is more relative, but oftentimes it's actually oriented like the measurements are with respect to the orientation like of a wall. So oftentimes these are done in boxy environments. So now that is kind of changing for reasons I'll talk about in the future.
Let me just, I think there's some more detail here that's worth
Yes,
pointing
go
out.
ahead.
On the left picture, we're showing two cells at different phases. These cells would be right next to each other, and a set of them would cover the entire environment, and that is a grid cell module. A grid cell module is a set of grid cells that all have the same scale and orientation, but of different phases. If in the middle one we have different scale, these are two different grid cell modules. So grid cell module A may have one scale, grid cell module B may have another scale, and they are typically arranged in a linear form and going to larger and larger scales. And then finally, two grid cell modules, when they anchor on an environment, and when a grid cell module anchors on an environment, like when it says, oh, I'm representing this room, it has an orientation relative to the room. And so two grid cell modules can have two different orientations relative to the room. And any grid cell module could orient itself to, vertically in one room and diagonally in another room. So we're looking at, in the left, we're looking at properties of cells within one grid cell module. And then on the right, we're looking at the properties of cells in two different grid cell modules.
Yes, yeah.
Cool.
Uh,
Are cells in a grid cell
uh,
module generally next to each other in the brain?
Right, they are. And there's a famous picture we've looked at several times here from a tank, I call it the tank paper, where he shows exactly how these cells are aligned. And he proved that they are literally, in a grid cell module, they're literally, the adjacent cells are adjacent, just like in this figure.
Yeah, and then so these are the three, or I guess the fourth one that I would add in that that is common in grid cell literature is something called gridness score, or GS. So you'll probably see that in some of the figures that I screen capture. That's what it means. But basically, how gritty the cells are. So how they calculate this is once they get the firing field, they mask out the portion. This one is easier to see. So that they take the hexagon, mask out everything else, mask out the middle portion, rotate it by 30, 60, 90 certain degrees, and calculate the correlation between the rotated version and the original version. So if it's very like regular grid-like pattern, you're going to have like a high correlation score. While if it's not well correlated, it will be very low. And the difference between the two is a grid score. So in this case, like in the very like grid-like cell, this score will be very high because after like 60-degree rotation, it will have a very high number. While in 30, it will have the very negative correlation. So the difference is much bigger. While in this one, the grid score is much smaller. And I'm mentioning this because I realized I guess that when I was reading like one of the original papers, I was like, how did they know that this is like grid cells? Like this is like purely a functional characteristics. And I generally come from like a more like morphological or like a transcriptomic way of defining cells. I was like, so how do they know to like poke the electrode here? And I realized that it's more ad hoc. It's like they measure these firing rates after sticking the electrode, and if the grid score was above a certain threshold, then it was characterized as grid cells. So I think the way they kind of threshold for grid cells is a little bit different by paper, but at least the original one, they consider any cell that was that had a score of 0.2, gridness score of 0.2 or higher to be grid cells, and
Right.
rejected the other ones.
But
So
it's even more complicated than that, or at least more interesting than that. So when they first started looking for, as you point out, Hojae, they're trying to find out what was the predecessor or the lead into a place cell, and so they looked in the entorhinal cortex. By the way, the O'Keefe lab looked there first, but they didn't find grid cells because they were looking at grid cell modules that were too big in scale. And so they couldn't find cells that responded to the small environment, where the Mosers came along and by chance picked another part in the entorhinal cortex where the grid cell modules were smaller scale, and they found grid cells. Now, you're right. What they were once I found sort of this grittiness, they looked for cells that were really gritty, right? That had this behavior. So they found, oh, we found these grid cells. But there are huge numbers of cells in the entorhinal cortex that are not classic grid cells. They might be directionally sensitive, so the cells respond when animal is moving in one direction, but there's still some grittiness to them. So you could have like a cell that was directionally sensitive, but also fired in a periodic locations along some dimension. And so that's not a classic grid cell, because classic grid cells are they're independent of movement and direction. They're just like this, you're at this location. So now they found all these different types of cells in the entorhinal cortex, dozens of different ones that are not classic grid cells, but you can then assign a grid score to each one, saying, oh, this one has some grittiness to it. This one has more grittiness to it. And of course, the classic grid cells are if they find those, and those are all very nice and gritty. So it's a way of not just saying which is a grid cell or not, but it's also a way of saying, hey, there's all these other cells that have shared some properties of grid cells.
Yeah, so also to go back to like Quietly's earlier question, are they next to each other in the sense that like are they like two cells kind of connected? I don't know if it's that close, just by but they're close enough that
they are in.
You mean this picture?
We can look at the tank paper
this
later.
Picture?
No, it's only in the tank paper, where they prove this in that within a grid cell module, the cells are they are laid out like a map in terms of they're immediately adjacent to each other.
Okay.
I can show you the picture later if you want to see it.
Okay. Yeah. So
but all these, by the way, all these other cells is remember like a lot of things, scientists will find, oh, we found these grid cells, but there may be only 10% of the cells, and 90% of the cells are not grid cells, and they don't know what they are, so they don't report on them. But they say, oh, but they're gritty, so they have grittiness to them.
Right, yeah. So yeah, that's another question. It's like, what percent of cells in the entorhinal cortex is grid cells? I'm not exactly sure,
I don't know either.
honestly.
But it's
Uh,
like you know, when Hubel and Wiesel did their first experiments on cats, they reported all these cells at this edge detectors and so on. That was less than
Yeah,
half the cells. More than half the cells, they had no idea what they were doing. And
yeah,
they just, you know, they'd say, well, these cells seems to be wonky, be spikes every once in a while, we don't know why, or these cells seem to be silent. So the same thing happens here. They find cells, they're looking for cells that are gritty, and then they say, oh, here's one that has some grid properties to it.
yeah. And it was kind of an interesting concept, at least coming from me, I guess. Like I would think that you would find something that might be grid cells first, and then measure the properties, but kind of backwards in some sense. For me, anyway. So so how these some of these properties can vary, like Jeff mentioned, scale varies by scale is actually organized anatomically, like so as you go from dorsal to ventral in entorhinal cortex, whatever that kind of means. Dorsal and ventral in this sheet doesn't really make sense, but it kind of in
Well,
general.
it
So
is
like
relative. It's relative to the head, right? It's
right,
it's like a dorsal fin on a shark. It's like towards the top of the back top of the head. And
yeah. I think for
ventral's the other
mouse it's
direction.
actually the it's consistent, but for humans it's like the dorsal. But I think I remember like the definition flips as you go from brain to spinal cord, because the
Maybe it's crazy.
yeah. Anyway, yeah, it's a little crazy. So, so I'll just have to take this figure for it.
We
But
don't. It doesn't matter.
basically,
It's only important if you're
yeah,
cutting into the animal and trying to find this stuff. If you don't need to,
right, yeah. And so basically, so like within kind of that anatomical region, the grids will have like small scale, and it'll go increase, and it will increase as you go to ventral. The So I think most people agree that this kind of scaling is like a geometric progression with about, I think that Moses reported, 0.42 because it was a square root of 2, which means that area, kind of increases by a factor of 2 for every module. Some other report like anywhere between like 1.3 to 1.7. But basically, when we say grid cell module,
this is not a continuous change.
it's
These
not
are
a
changes
yes.
like you go from one grid module at one scale, then it jumps to the larger, and then it jumps to the next one.
Yeah. Actually, I think this bullet point is incorrect, but so I think the original there's many consensus or opinions on how many modules of these actually exist in the animal. I think one that I heard first said 4 to 5, but now other papers have said like up to 10, maybe. So, but I think right now the general consensus is probably somewhere between 3 to 10. I would say.
This is a very important number.
Yes, we
I
just.
don't know
I
if are
feel
you
like we
going to
just don't
cover that?
know.
Why that's important?
Go ahead. I'm not gonna. I was gonna move on to the next property.
I don't know if you know all this stuff, Hojae. So it's, as I said, it takes months to get it all.
Yeah.
So if we want the way to get unique representation, initially another people proposed it, and we included it in our frameworks paper, is that if each of these grid cell modules anchors independently, then the combination of which cells are active in each grid cell module becomes unique. And this is where the idea of unique locations comes from. But to really mathematically, to get a unique location like we would like, from a theory point of view, you need more than 3 to 5 modules. You want at least 20.
Yes.
And also you wouldn't really want them at very, very different scales. That wouldn't work too well either, because if you're in a small environment, the big scale doesn't really tell you anything. So this is always a problem that remains if we're going to find unique representations of space. You need to have you need more than 3 to 5 or even 10 modules like this. And so I'm going to propose later how I think this might be occurring. But we were so excited about this because it's a really cool idea that you can combine a bunch of grid cell modules and you get this unique location, even though each grid cell module is not unique. And but the numbers never really worked out very well for us. So
Yeah.
but I have a potential solution to that.
Right.
Yeah,
Did you
maybe
did
it's
you
just
actually
worth
go?
commenting on. Oh no, go ahead, Hojae.
Oh, go ahead. Go ahead. Go ahead.
I was just gonna say it's worth commenting that orientation can also contribute to that. The orientation differences, the coding differences. Like basically,
Right.
you want
Yes.
kind of looking at the maybe if you go back a slide, sorry, Hojae.
You're right. I didn't
Yeah,
know.
this one. Like basically, you can imagine that with shifts in phase and orientation and/or scale, you can basically get sort of interferences where you have different grid cells are all firing at one particular location, but they don't co-align, they don't kind of interfere at other locations. And that's what kind of gives
Right.
you the unique readout of a location,
So-
despite that a single grid cell or even an entire grid cell module is cannot uniquely define a location.
So one of the things we talked about, and I think we talked about it in this group, but prior to this group as well, is that you could have multiple grid cell modules at the same scale, as long as they have different orientations and different anchoring, as Niels pointed out. And but then you'd have to have a bunch. And I couldn't
Yeah.
find any evidence that there was a bunch of grid cell modules stacked on top of one another at the same scale. And so, that was a problem too. So-
Yeah.
I have a question.
Yes.
When we're saying the word unique, but what is unique? Like millions, billions, trillions,
Well,
every
that's
atom in the
right,
universe?
right. So let's do the math here on this. Let's say a grid cell module, a cell represents 1/10 of the area of an environment, right? So maybe you could say with a single grid cell module, I would know where I can only know where I am 1/10 of the time. Like it's not unique, right? Because the cells fire over and over again, different places. So, but a single cell might represent 1/10 of the area. If I had two grid cell modules that were not exactly the same, then it'd be 10 times 10. I could represent a unique combination of the two would be 100th. So it's basically 10 to the nth power. If I had 20 grid cell modules, it'd be 10 to the 20th. That's how many unique locations I could represent, which is a very big number. So that's sufficient for us. But 10 to the 3 or 10 to the 5, probably not, And so you would want to have a whole bunch of these things so that the combination would be unique and that you wouldn't have false positives and false negatives and things like that. Does that help you answer that question? You look confused.
I was just running some numbers. So so 10 to the but is 10 to the 7th enough, or the 10 to the 20th
I don't know.
is enough?
What's enough? I don't know. Maybe.
That's what I was asking,
well-
like 21, because there's math you can do where you can have 5 grid modules that will give you 21 million unique codes.
Well, how
And
do you do
so,
that? How'd you get to that?
The phases have to be, the phases can, if they're co-prime phases, like 3, 4, 5, 7, 11, they will align perfectly.
Remember, in cells and neurons, there isn't any kind of thing like the coding we have in computers. I mean, if I have a bunch of neurons and 9 of them are the same and one's different, that's not unique, right? These are sparse distributed representations. And so you can't say, it's not like, oh yeah, one cell different will give me a very, very large number. That's what we rely on computers, right? Every bit makes a big difference. So you can just say it's 2 to the n bits, and that's how many representations you have here. That's not the way it works here. You have to, the cell combinations have to be fairly unique. And this is what you get with sparse distributed representations. We could spend a lot more time on this, but I think
Well, the dimension of uniqueness I was talking about is the phase. As long as the phases are co-prime with each other, 5 different modules with 5 different phases that don't overlap, like that are co-prime, that is like it's rare for them.
A grid cell module doesn't have a phase. It's the cells within the grid cell module has a phase.
Right.
So
Yeah, the cells
think
within
that-
the grid cell modules. If those are
You're right, so-
co-prime and you get 5 of those, then you can get 21
No, I
million
basically-
separate.
But I think that's just point. Like all the cells in a grid cell module have the same phase, so there's no amount of kind of interference between those. They'll kind of
right.
always fire at different times and kind of consistently.
I think, well, okay, I might say something a little bit different on the slide, which is that the cells, like within the module, the grid map, I can just read what I said here. Within modules, the grid map is rigid and universal, i.e. scale, orientation, and phase relationships are preserved across environments.
I don't think
as
that's
in,
right. Is that right? I didn't think, I mean, the cells within a module have the same scale, orientation, and phase. But in a different environment, they have different orientations. I mean, the whole grid cell module can be rotated differently in another environment.
yes.
So within a module, there's a rigid relationship between the cells.
Yes, but there are cells of different phases. So as in, this orange is slightly, translated from the blue, by a little bit in this messy picture. So within a module, yeah. So this is still within a module. The orientation, I think in this case, the orientations are, I think, the same. It's a little bit hard to tell, but at least the
Yeah,
phases are different.
I think these are all the same module. These cells, aren't these cells?
Yeah, in the same
Yeah.
module, like 3 cells
All
that are
the-
kind of nearby each
All
other.
cells within a module have the same orientation and the same scale.
Yes.
Always,
Yes.
always. There's no change in that.
Yeah, but the phases.
The phases just represent this cell responds to this area, the next one responds to a different area,
And the
those-
frequency, which is basically the scale, is going to be the same for all of them. But yeah, I
Yeah,
mean,
yeah.
maybe it's worth just pointing going back to your comment, Tristan, that these are grid cells in the entorhinal cortex. We don't know if the exact same properties exist in kind of whatever is giving us location path integration in cortical columns. Like maybe that is more of a code based on, as you're kind of saying, like different phases that then interfere.
But I don't think so.
Yeah.
I think it's extremely unlikely. So I'll state that. It's possible, but I don't think so.
Yeah, well, Jeff is
give
going
my
to go into more
justification.
detail about. Yeah.
Um,
So I think what Tristan was saying, it reminded me of this visualization where, you have grid cells of the same, this is from one module where it's the same spacing, but different phases. But it's only this location where all 5 kind of fired together, while in other locations, only 2 of them or 3 of them, might do so. And that might be how it might be giving like a location.
these are different grid cell modules, right? These are not cells in the same grid cell module. These are different grid cell
These
modules.
are, no, same module because they have the same spacing and orientation, but different phases.
Then, but if because
But they
no,
don't have the same
no.
orientation.
And they don't have the same, no, otherwise you wouldn't get this pattern. I told you this is a very confusing field.
Okay.
This is, you see what they're showing here is that there's a bunch of grid cell modules are firing at the same location. Those those grid cells are going to be in different modules. And there's a module, the different color cells are in different modules and they have different scale and they have different orientations. And what this is showing is that even though each module is not very unique, each module cannot tell you very much detail where you are. If you look at the combination of the modules, at any point in time, only a set of one cell in each module will be active, and therefore that combination is pretty unique. So these have to be in different modules, is my understanding.
Right, and so this is what I was trying to say is you only need 5 modules to generate 21 million unique spots where they all align.
Well, I don't see that. I don't know how you get that number, Tristan. I don't think that's right. So I'm not sure what math you're doing to get to that.
Yeah, but that's what I was trying. That's what I was asking. Like, because if we're saying that there's 10, then mathematically it is possible for 10 modules to reproduce.
I think you and I are thinking about it differently because I don't think 10 is sufficient. But 10 doesn't get you that kind of numbers.
I mean, I can share
for what it's
something in
worth,
another
like
meeting.
this is showing something different. This is showing like, yeah, kind of what Jeff's describing, like the core idea behind how different grid cell modules do it. Because these have the same spacing, it's the same scale. They actually, it's debatable. You can't really say whether they have different phases, because clearly at one point they're all aligned. So in that sense, they're in the same phase, but they have different orientations. And so that's what's giving them this like only 1 point of interference. If they had, different spacings, then within a module, which if that was a thing, or like let's say within a cortical column, whatever, then yeah, that could maybe do something like that. But it's not really the phase. It's it's more the spacing that needs to differ.
The spacing or the orientation?
Or the orientation.
Those are the two things that give you.
Because the phase is what tiles space. And so like they're going to have different phases, because otherwise you're going to have gaps where no neuron fires.
Here's another way to look at this. I'm not addressing your question directly, Tristan. We can try to figure that out later. What grid cells give you, the important thing they give you is they don't give you unique location, but they give you the ability to do path integration. So that's a property that's we mentioned before. If an animal's in the dark and it moves, the grid cells fire correctly in sequence. And so there has to be, for that to work mechanistically, there has to be some sort of structure to them that's repetitive. And I don't know how to describe it other than just to say that. So you get this ability to give a movement vector. The animal says, I'm moving in some direction, and the grid cells change. Which grid cells in a module change? The adjacent one becomes active, and another one becomes active, and another one, and then they repeat over and over again. But it gives you that ability to do path integration, and therefore this regular tiling of the space is actually essential for that. The mechanism wouldn't work if within a grid cell module, all the cells have to have the same spacing and the same orientation, and they have to be just separated in phase. Really, phase meaning just one fires here, the next one fires here, the next one fires here as you move through space, and then they repeat again. The property of this grid of being a triangular lattice is essential for the mechanism to work, and we can't get away with that. We can't get away from that. I mean, we can encode to whatever we want, but neurons can't do that. I don't know if that helps, but I'm just trying to make the point that within a grid cell module, all the cells have the same spacing and they're arranged and they all have the same orientation, and you take the lattice that one cell responds to and you shift it over, that's the next cell, and shift it over, that's the next cell, and shift it over, that's the next one.
Probably need a paper to actually find that. I mean, if so, I mean, we will solve this kind of uniqueness problem if, we only allow the scale to be different across modules, but allow for phase and orientation to be different within a module. Then, like things like this can happen.
The uniqueness requires multiple grid cell modules.
Yes.
And number of grid cell modules, where the grid cell modules differ in either scale or orientation.
Right.
And they on any particular environment that they remap, right? The orientation and which cell is active in which environment, they have to remap. So it's like the same grid cell modules, but they remap randomly, let's say.
Yeah.
I don't know how to describe it better. It's so damn hard.
So my original question was, so what is do you have an intuition for what is unique enough? You said 10 to the 20 is unique enough, but like 10 to the 10 maybe isn't.
Well,
is it somewhere in there?
I don't know.
For our needs.
It's just a great question. I mean, when we want in our learning modules in Monty, which is equivalent to cortical column. So in a cortical column, what you'd want is that there's a representation of location in layer 6. And that has to be unique enough so that I'm not going to confuse that location for a location in other objects or locations on the same object. So what is that? It depends on how many objects I learned, and it depends on what's my tolerance for making mistakes or false positives.
Yeah.
So I don't have an answer to that, other than you want it to be pretty unique. You don't want to have mistakes, I think.
Yeah, there is. I actually posted about this on Discourse today because someone was asking about it. In the paper that Marcus Lewis was first author of in 2019, there's a fairly extensive analysis of how you vary the parameters, like the number of grid cell modules, how many cells are in each grid cell module, things like that. You get kind of a change in the capacity to learn different objects before they start interfering with one another and causing the system to fail. So yeah, if you're interested, Tristan, I can link you to that, and it goes into more detail.
Here's another thing that's interesting. When an animal first appears in an environment and it doesn't know the environment, or it doesn't recognize it, maybe it just hasn't gotten enough information to know where it is. And the grid cells anchor in one way. And it's sort of, I think it's more of a sort of a generic way. It's like, I have to check on this. I'm not 100% certain about this, but there's a couple of things happen. One is the grid cell modules seem to expand their scale a little bit, like I'm not really sure. And then they also sort of anchor in a way that's not unique. So it's more like a generic room. Okay, this is a generic rectangle. But then if I recognize, oh no, this is the particular rectangular room that has the food in this corner and the little toy in that corner and the window over here, then they re-anchor to form a unique representation. So it's like this sort of a non-unique default anchoring that is like generic space, and then becomes, oh, I know what it is. It's this particular one, something like that.
Is that non-unique re-anchoring with respect to the body? Is it like egocentric?
No, it's always allocentric. It's always respect to the environment.
It has to be with respect to some objects.
No, it just could be random. It could be the default. Well, I guess I see what your point, Rami. I don't know. I guess I see your point. I don't know. I think, I mean, I don't know if there's any experiments where you put the rat in like a featureless void.
Yeah, I'm not sure we found a paper that explores this, but it would be interesting. Like, yeah, that
I think it's
this
always
kind of in general, the animal knows, I think, what environment it is in. It's
Or-
kind of
or
right
it-
away.
or
It's hard
or it
to
knows
yeah.
it knows it's in a rectangle. It may not know which rectangular box it's in.
Right, but
So-
normally, I think this is deliberate. Normally, they don't obscure information in the room. Like
Right.
there
but
will
they've done
be like unique things on the ceiling and stuff like that, and that's just considered part of it, that the animal knows it's in that room.
But I think there have been experiments where they do it, because I do remember these experiments where when the animal doesn't know what room it's in, it forms one representation, but when it knows what room it's in, then it anchors, and then it knows. So it brought up a very interesting thing about these experiments. These experiments typically are the rat in a maze or a box, and the top of the box is open. And so if you looked up, you'd see the room of the experimenters. You'd see the lab. Sometimes you even see the experimenters themselves hovering over. The rat would see them, but they ignore that and they say, "Oh, this is a featureless box," and so the rat's in there. We didn't put any features on the walls yet, so he doesn't know what room he has no way of knowing. He has no input, which is bullshit, because the animals can see the room above. So, but there have been some experiments where they completely seal it off, and then you see this property where the rat sort of, I believe, I have to go check again, it's been a long time, that it forms a sort of generic representation of space based on, is it a box or a rectangle or a circular environment. And then when it becomes a particular circular environment or a particular rectangular environment, it re-anchors.
Yeah, well, I mean, that would be great if there were some studies. I'm curious, Hojae, just to kind of preview what's to come next. Did you come across anything like that when, in the limited time you had to look?
You mean like the wall being covered or not?
Yeah,
I mean,
as in
I know that
like
I don't know
an
about
experimental
ceiling.
setup where like the animal would be unsure what room it is, and then they're measuring grid cells, and then it suddenly
There are
recognizes
lots
that
of
it's a different
experiments.
room.
There are lots of
But
them.
I feel like often they're
Yeah.
focused on place cells and place cell re-anchoring. I
I
don't know
so-
if they specifically look at what the grid cells are doing.
I'm pretty sure.
Yeah.
I bet you if we asked Marcus or Subutai, they'd remember.
Yeah. So okay.
I mean,
So
the whole idea of re-anchoring is, I remember they would take a room and they would hide, the room would be identical until they put like a white placard on one wall. And then the animal would re-anchor to that white placard. They put a red placard on another wall, the animal would re-anchor differently. So there is this sort of anchoring based on visual clues that tell the animal which is which.
definitely, yeah. Yeah, so if they color like one side of the room differently from the other, it's that can cause re-anchoring. Yeah. The other, gosh, thing related to like unfamiliar environments is that oftentimes they say like, okay, we waited 4 days until the animal got familiar with the environment and then measured, the grid cell whatever activity. And okay, so this is a slide that I didn't add in. Another, and also like a secret property that I didn't add in here is field size. So basically the size of the circle is another kind of property that is sometimes mentioned but not often explored enough. And that is basically, like what's the radius or the diameter of the firing field. And what they found is that in unfamiliar environments, the field size would temporarily increase. So if you take a mouse and go somewhere, the field size will temporarily increase, and then over time it will decrease, which also indicates to me like, okay, if the field size is decreasing, that means that there's potential. I don't know. That means that if there's like if there's enough number of grid cells, then basically the resolution of our location is getting higher, I think, and also by that the number of unique locations that we can identify gets higher. So the more
No, well,
I'm not sure if that's true.
That's
but
not true, because think about it, the number of grid cells hasn't changed. So imagine these grid cells perfectly tile the space. So every place in the environment, at least one of the grid cells is firing. If you make the grid cell field larger or small, if you made it smaller, then there's spaces in the environment that wouldn't respond at all. So it's with making them larger, you're just sort of introducing uncertainty. You're just saying, oh, well, I'm not sure from here or here or here or here. But it doesn't increase the resolution of the environment. That's dictated by the number of grid cells there are.
Yeah.
If you don't increase the number of grid cells, then you can't encode more space. You can't have more locations.
Yeah, maybe not resolution, but certainty. Kind of like you might think you're in multiple locations, like kind of multiple hypotheses, to use Monty terminology, while the field sizes are large. But then as they narrow, those hypotheses kind of collapse into a smaller number or one.
Here's another thing that I think they showed. You remember I mentioned earlier, if the animal's in the dark, the grid cells do the path integration, right? But and I've talked about this too, you can experience this yourself. If you're really in the dark and you start moving, you get more and more uncertain as to where you are as you move. And then if you then find a clue, like if I'm in the dark and I touch a corner of a wall in my room, oh, I know where I am, then you're exactly anchored again, exactly in space, right? So I speculate, I don't know, but I speculate what's going on as you path integrate and you don't have any visual or any other clues to tell you where you are, those firing fields will get larger. It's just like, yeah, you're path integrating, but you don't really know where you are anymore. It's some blobby area. And then you get a clue, and then they narrow down again. That's how I've always suspected that happened. I don't have any evidence, but that's what it feels like.
Right. My phone just beeped that it's been 1 hour, so
Oh, God.
and I'm only one third of the way through. So okay, so let me see if I can pick up again. Okay, so kind of went over this slide. Grid cells and other animals. Okay, it's a simple one, and also I wanted to kind of put one of the most important ideas of where TP came from. So one, grid cells found in mouse, rat, macaque, bats, human. Human only by fMRI, because you can't just poke an electrode into your hippocampus. Not recommended. Bats might seem random, but that's actually because they wanted to see how if there's a grid cell activity in animals that navigate naturally in 3D space, aka by flying. And yeah, so the main idea that we had apologize just saying this honestly, is that so we're combining kind of two types of experimental evidence. And one, by fMRI, we have some evidence that there's grid cells in the human entorhinal cortex. And then there was another paper was that there seems to be grid-like activity in the somatosensory cortex of mice. So combining these together, it's not really a stretch to say that there's probably grid cell-like activity in somatosensory or other cortical areas of human. I think
That's all true, but that's not how it came about, Hojae. What it came
Okay.
about is we logically deduced that cortical columns must have a representation of location of the sensor input. We logically deduced that there has to be path integration. You just move your finger and you anticipate what you're going to feel.
Yeah.
We knew this had to happen on a local basis. It had to happen of every patch of your skin. It's not clear that it has to happen locally in the visual cortex, but we said, yes, it does. And then we said, how could a cortical column represent a location with path integration like that? And a priori, I had no idea how neurons could do this. It's just like, how could they possibly do this? I have no idea. But then we realized that grid cells did something similar. And so we studied grid cells. And we realized, oh, grid cells are this really complicated mechanism that does exactly what we want. So then we took a guess saying it's unlikely that cortex evolution came up with a different mechanism to do the same thing it was already doing in the entorhinal cortex. Much more likely, it took the same mechanism and repurposed it in the cortex. So it was pure speculation. We didn't know about the evidence of grid cells in neocortex. We did not know that. We had no evidence for that at the time. And we said, you know what? We're going to guess that there's going to be grid cell-like mechanisms in the neocortex, in the lower layers, throughout the cortex. And then afterwards, we learned that there was evidence for this. But it seemed a fairly safe bet to say, like, this is so damn complicated. It's immediately, it's in the same cortical sheet, it's just further over, in the same sheet of tissue. It's almost certainly that whatever evolution discovered for the entorhinal cortex is going to be used in cortex as well. And then we found evidence of that.
I see.
It's not, everything you
There
said
you have
is
it.
true. It's just the order in which it happened.
Yeah. Okay, so and then I went over this. Oh, by the way, these two pictures are from the Numenta HTM school. I think somebody created like a Jupyter notebook of like how you can kind of like move your mouse over, and then this like tiny little mouse would actually like move over your screen, and like it will be colored by this kind of circular grid cell firing fields. I couldn't find the actual GitHub repo. Maybe somebody else might, but I did find a YouTube video of it. Anyway, so all these, and I'm glad that I put this asterisk. Actually, the story may not be this simple in terms of how grid cells give rise to like place fields or unique locations. The idea generally that was kind of proposed by the Mosers was that if you have grid cells and of different modules and you kind of stack them together, depending on how they overlap or interfere, that would give unique fields. But I feel like it's a little bit more complicated than that. And then there's also the question of
I
like,
didn't know it was the Moses who did that. I thought someone else did it, but I'll take your word for it. If they might have done that.
yeah,
Yeah,
At
like
first-
Elif feet and
Right. I thought there was.
yeah.
So
I mean,
know
I think
that, so.
honestly, people's like opinions are changing. I feel like even within the past 10 years, so it's a little bit hard to say. And
By
then
the way,
there's
the
also a question of
research-
like,
some of the researchers in this field are very competitive with one another. So they.
okay, so maybe I shouldn't say.
No, it's just, I'm not going to say names, but I do know there's a lot of counterclaims and claims.
Yeah. It's very interesting. This field is very dynamic, actually. I thought this was kind of like stalled in some sense, like this textbook knowledge, and then I realized this is very not textbook knowledge. We're debating a lot of things. And Jeff, this is my only mention of the word oscillator interference, which is one of a couple of models that tries to explain how grid cells are firing fields are generated. Because the actual question of like, okay, if we kind of if we take this hypothesis that combinations of grid fields can give rise to place fields. Then the question is, where are these grid fields coming from? And there's a couple of theories, and sadly, that's all the mention about it. I do
Yeah.
think, yeah.
Go ahead.
Anyway, so I will get through quickly so we can actually get to that part, or you can explain
Okay.
more about
I mean, it's
oscillatory
on its
interference.
own. One can just ask, how do grid cells get this property? What makes them fire like this? And there are two basic theories. One's called oscillatory interference, and one's called continuous attractor networks. And I believe the right answer was exposed in a paper that said it's a combination of the two. So I'll talk about that later.
Okay. And I think there's like 4 new meta videos on hybrid models of grid cells, OI, oscillatory interference, and continuous attractor networks. So if anybody's interested, there's like at least 4 videos of that from like 5 years ago, something like that. But there'll be an update here as well. Okay.
Yeah, I wonder if, I don't know
yeah,
if
go
this
ahead.
would be a good time to swap over just for a bit, at least, to Jeff, what you were going to present, and then we can see how much time there is for the rest of this Hojae at the end, and then
Oh yeah, that works too. Yeah.
since you were going to kind of talk essentially about how grid cells emerge, Jeff.
Yeah. Maybe that's a good idea. I mean, we could spend weeks going down the rat hole of grid cells. I mean,
Right.
it's unbelievable. But I think I could bop up a level to make it more applicable to Monty here.
Yeah.
Sounds good.
So
Maybe
if
can
you
I
don't mind
do
that.
just like one recent work that
Sure.
may be interesting? This polyalums. So I think this 2022 paper was pretty interesting in the sense that, so what they did was that instead of taking this tetroid, they took the new neuropixel electrode, stuck it into the mouse, and measured the activity of grid cells within a module across time, and then they so they have a population vector. So basically they measured about like 500 to 1,000 cells at the same time. So each time point has a vector of the number of neurons that they have measured. So in this humongous 500 dimensional vector, they the dimensional reductions of PCA and something called UMAP to make this visualization, and they found that it seems like the activities of these 500 to 1,000 neurons from the same module seem to be forming a toroid. And what
Isn't a toroid just describing the fact that they repeat? I'm confused by that.
I'm not that far, yeah, but I think the most interesting part was that when they look at the grid cell firing in this regular room of box, you get the hexagonal pattern, and the same kind of equivalent pattern in this toroid, kind of this indicated by the black dots. And when the mouse moved to a wheelbarrow like environment in 2D, the firing patterns have changed, obviously because the environment's changed, but in 3D it hasn't. And I'm not saying that I think this is kind of
interesting.
Okay, yeah.
I'd like to read that paper if you post that one.
Yeah, of
just
course.
a general comment on this area is that grid cells seem to represent 2-dimensional locations, or location in 2-dimensional space. And so then the natural question was, can they represent 3-dimensional spaces? And they did a lot of work with bats flying in large environments. I mean, huge, these big tunnels and stuff. And they can record from these cells while the bat is flying, and it's amazing. And what they found that the grid cells still seem to be they fired in particular locations, but in the 3rd dimension they lost their grittiness, and they were sort of random looking, and their size changed, and so a whole bunch of weird stuff. So the last time I looked at this field, it was completely uncertain as to whether what's going on in 3D.
Yeah.
Is that so? This may relate to that. Is that what this might relate to that?
Well, I was going to relate to re-anchoring as in there's this particular statement that I was very confused about, which is grid cells form a metric space. This statement. And I was like, at first I was like, okay, this makes sense. And then I realized, okay, but if we're doing all these re-anchorings, it's not really a global metric space, is it? I mean, just the fact that we are re-anchoring and there's many ways that we can re-anchor means that
But,
not
individual,
best, it's like a local.
You're confusing two things there. Within a grid cell module, once it's anchored, it's metric. And
Yes.
meaning that I can start anywhere and move in a certain direction and I get the same results, or I can do this path integration, come back to where I was. I mean, it's got this regular structure to it. That's independent of re-anchoring. It's always going to be metric, but it's just a property of the cells as opposed to where it is in the world. So
yeah, so I guess I'm still kind of sorting through it out of my head, but yeah, I think one of the most striking things was that these patterns can be, steered, compressed, whatever, in different geometries. Or there's this crazy, like, I think you probably saw the trapezoid one, but I'm not sure if you saw the hairpin maze one.
yes.
You still have some, like grid periodicity, but it's very dependent on the environment geometry. And I thought this one, this 3D one was kind of interesting because I think the authors were trying to say that there's some environment geometry invariance to this. That said, like I don't know if this, they only tested on 2 different environments, and none of these are still natural environments, I would say. So
I think why don't you just post that paper and
yeah,
we can look at it and see its relevance to the issue of 3D space, and which is an important
sounds
topic
good.
for Monty, right? We
Yeah.
talked about in Monty, well, if grid cells really are 2D, and there's evidence that we've come up with, just observational evidence that we learn 2-dimensional maps, even of 3-dimensional objects. Talked about faces and dice and things like that.
Yeah.
But we also said for Monty, it doesn't really matter. Why don't we can use 3D or ND? So it's a very interesting question from like, what does neuroscience do? What can we do? Do we have to do what neuroscience does? Hopefully not. But it would be nice to understand if I understood how nature really represents 3D environments or spaces, that would be helpful for us to figuring out what we might want to do with Monty, if what we're doing is sufficient. Okay.
Yeah, it seems this is more kind of like you say, Hojae, kind of about what do you do in terms of keeping the metric space consistent rather than necessarily helping with the 3D thing. But
I see.
but in that sense, I feel at least not too worried in that, like, I think to do what we want to do, which is, recognize objects and make predictions, it just needs to be consistent locally. And I think that kind of fits as well with, like, we experienced this ourselves when we were doing the TBP Olympics at the retreat, that, like, if you were to close your eyes and try and, like, feel, an absolutely enormous object and, like, path integrate over the whole thing, you'd eventually get lost and confused about what you're sensing. Path integration is pretty much always going to be noisy over, like, certain distances. But it's more just that, like, within a certain region, it's like, okay, yeah, this feature is above this feature, this other feature is to the right of that feature, and things like that. That's the kind of key thing. And so even if it's like re-anchoring because, okay, this is a new object, it still gives us that kind of what we want of basically integrating movement.
Another thought that I had with TP Monty, remember, like 1 month ago we were talking about 3 streams in in Monty, their behavioral stream, morphological stream, and the feature stream. Maybe so we're kind of separating out the features from the morphology. If these are kind of like recorded not in the space that we are observing, so like we observe things in 3D in habitat, and then we kind of put that in a location in 3D, like this, if this kind of representation basically gives an additional dimension, that might give us a little bit more representational power to encode something more generic, possibly like a morphological, like a generic class of morphological model. And somehow, like, the combination of the morphological model and the features give a specific instance of a particular mug or a particular, like
Well,
a TP mug versus
I
a green.
think
Yeah,
some of that
this
is,
is, I think that's too far.
but that's right. But we do that. We're using that during the temporal memory mechanism. Right? We're defining many columns are defining the morphology of an object, and then you can form unique representations of that morphological object, not by changing the location space, but by changing the encoding of the feature location combination. These are all very complex topics, and they take a long time to absorb.